Właśnie. O, jest takie super. Zamknę drzwi, nie?
Okej, wybiła godzina 14, także myślę, że możemy zacząć. So, ladies and gentlemen, welcome the afternoon session. It is my honor and
To be together with you and during this time. Be a very interesting time together, very valuable. In context of the valuable information, I would like just to contribute today with a stone that can be as a milestone between present and future in context of nanomaterials knowledge. And before starting my presentation, I would like shortly just present my background. Generally, my scientific background is divided in two parts. One part, it is before 2014. Second part, it is after 2014. My experience in my country, Moldova, it was started with the work in such cosmetic company called Viorica Cosmetic. From this company, I just received the proposal to go to the university and join first step of my experience at the Industrial and Ecological Chemistry Department at State University of Moldova. Then I passed the second step. I passed the second step, and the second step, it was my master's degree that I received in 2010 in technology of cosmetics and medical products. Then I moved to the another cosmetic company. Start the PhD study from the. But later in 2004, I suppose it is fortunately, I received another invitation from Professor Buszewski from Poland to start a new PhD, new topic, new life generally in a new country. So I closed my step here, come to Poland, and in 2018, I received a PhD degree, and the title of my PhD thesis, it was Silver Nanocomposite as a new safe antimicrobial agent. Currently, I'm associate professor at the same university, Nicolaus Copernicus University in Torun. My research field is regarding nanomaterials, synthesis, characterization, In step of the characterization, also application, I'm interested also in whole cytometer technique, which is technique that is very well known in diagnosis, but less known in basic research. Also, I'm trying to use such nanomaterials as a potential antiseptic drug. I'm trying to introduce such nanocomposites in the ointment formulation and verified for their effectiveness. So let's start with the topic of my presentation today. Of course, we will be regarding the bio-nanocomposites. In this context, I would like to debate today a few aspects. Firstly is aspect is sustainable development. Second, it is employment of the interdisciplinary approach. Then the consequence of such interdisciplinary approach means similarity and dissimilarity regarding the property. Also the impact of the concentration. And finally, I would like to discuss about the application of such nanomaterials in vitro and in vivo studies. Let's start with the question, why bio, why recycling, why sustainable development, and why bio-nanocomposite? Generally, when I start to work with bio-nanocomposites in 2014, in our group, firstly, we tried to use microorganisms isolated from the soils. Soil, it was as a source for us for such bacteria that were used for the synthesis by biological method. I have experience only in biological method and not chemical one. But as time went on, the increasing of the ecological problem just encouraged us to search new sources, new solutions for the present needs without compromising the future generation. And of course, our group was focused then on the using of the milk and whey as a byproduct, as a result of the recycling. And we tried to use firstly such sources for the, it was of course in 2016, and firstly we tried from one side to isolate protein that then were combined with the metals and obtain with metal proteins. This is not really my topic, it's topic of my colleagues, but also partially I'm involved. But from another point of view, we use such sources for the isolation of lactic acid bacterial strain that then we are using for the synthesis of bio-nanocomposites. Today, my topic will be mostly focused on the zinc and silver nanocomposite synthesized by biological method. So, all of these compounds about which I'm talking today were fully characterized by us. After deep characterization, we deduced that such compounds, such nanocomposites, such bio-nanocomposites, are consist from the metallic core and organic deposit, and for us, they are complex structure, not simple one. When we will discuss about metallic core only, we will mean nanoparticles. When we will discuss about metallic core and organic deposit, we will mean nanocomposites, means nanocomposites synthesized by biological method, with another word, bio-nanocomposites. Using such interdisciplinary approach, and here I'm meaning not only designing of such complex method for the characterization, but also I'm meaning planning of our work from the beginning to the end. Firstly, searching for the new source that can be eco-friendly, available, and inexpensive. Then use such source for the synthesis. Pass the next step, physical-chemical characterization and application. And by using of such approach, we were able just only after that to conclude such hypothesis. My talk today will be focused on 3 types of silver nanocomposites. 1 type of zinc oxide nanocomposite synthesized by 2 different bacterial strains, lactic acid bacterial strain, Totally, I will discuss about 6 nanocomposites, silver nanocomposite, 2 nanocomposite of zinc oxide nanocomposite. The method of synthesis, the using of side bacterial strain now are in the process of the patent preparation. So, how I said before, our nanocomposite consists from 2 parts, metallic core, organic part. How we deduce this, how we can check screening for the first step, the presence or not of organic surface that is covered this metallic core. Here I have presented 2 types of silver chloride, 2 types of silver nanoparticles that were separated by me from the same methodology that was basically before our method for the synthesis, but trying to use side bacterial strain in such medium, we met some problem. Then I was, of course, encouraged to develop new method. So we use in this discussion hybrid of them and this nanocomposite separate them. Let's start with the elemental composition and how we can see the differences between silver nanocomposite, pure silver nanocomposite, silver chloride nanocomposite, and hybrid of them. This is first step that it's showing us the differences between elemental composition regarding the organic surface. Second part, it is FTIR analysis that is showing us the presence and functional group in our system. Firstly, we can see the differences between organic surface, between the presence of organic or of functional group in the organic surface of pure silver, chloride silver, and hybrid of them. Then, secondly, we can observe differences between presence of functional group between the bacteria that were involved in our synthesis. Another aspect, this is metallic core, how we can differentiate such nanocomposite when we discuss about silver chloride and pure silver nanoparticles and hybrid of them. Of course, transmission electron microscopy, this is good technique, very nice, powerful technique for the identification and proving of the presence of metallic core. How we can see here in all cases are presented the metallic core of silver nanoparticles. In this context, we are talking about nanoparticles and not bio-nanocomposite, because transmission electron microscopy will see only the metallic part. What is next, and a very interesting aspect, because based on the electron diffraction pattern, we can differentiate such components, for example, nanoparticles, silver nanoparticles, pure one, and silver chloride, because in a different case we will have different results. Moreover, using such technique, it is possible also to investigate the size of nanoparticles. And I would like to underline once again, we mean here the size of nanoparticles and not size of biocomplex. So we observe that in case of hybrids, we receive higher size of nanoparticles. The lower size of nanoparticle was observed for the silver chloride. One aspect that I would like to specify here, it is the fact that the X-ray diffraction method, it is the best method that can be used for the differentiation of such complex, especially when we have silver nanoparticles synthesized by biological method, because generally, unfortunately, in literature, can see such results that they are presented such kind of the spectra or such kind of the spectra, and they are discussion in context of pure silver nanoparticles. And this information is a little bit erroneous because it is very well demonstrated that some signal are characteristic only for the chlorides. Some signal are characteristic for the pure silver nanoparticles. When we will have such compensation, we will see the signal that are coming from both pure silver nanoparticles and also silver chloride. And then we can say not about the synthesis of pure silver nanoparticles or silver nanocomposites, but the synthesis of hybrid of them. And this point should be taken in consideration for the future, because not so much researchers are taking this in consideration when we speak about the biological method. So let's move to the characterization of whole nanocomposites. And here we can design a few methods. We can use atomic force microscopy, we can use transmission electron microscopy, scanning electron microscopy together with transmission and scanning and transmission electron microscopy using various detectors. Firstly, we will see how the morphology differentiates from one nanocomposite to another. In this context, we talk about the nanocomposite, not only nanoparticles, because we see the morphology of whole nanocomposites. Then we can identify the size of nanocomposites, so we can remark here it's more higher than in case of nanoparticles. This is proving once again that our metallic core is covered by the organic surface. Another aspect, it is another combination. It is using such approach, scaling transmission electron microscopy with several detectors. Here, for example, here we have zinc oxide synthesized by the biological method by two different bacterial strains. Firstly, from the same place, we can differentiate the morphology, then we can differentiate the bio-nanocomposite size, and also using bright field detector, we can differentiate and prove the metallic core of such nanocomposite. If we will go through this and we will go through high resolution, we can see and we can remark such dispersion of metallic core of zinc nanocomposite synthesized by biological method, and we can see how this metallic core is dispersed in organic matrix, and this observation was found from different places of our sample. Using secondary electron, we can see how this nanocomposite looks. When we use different detector, we will see how this metal is dispersed in our organic surface. What I want to remark here, we did not observe some differences between the nanocomposites synthesized by biological method, but let's compare these results with the chemically synthesized one. Here we can observe such metallic core nanoparticles. We see the size of nanoparticles. Here we see how the nanoparticles is dispersed in organic surface. And we can see here maybe not dispersion, but cutting of such nanoparticles by organic deposit one by one. This cutting is take place in different way than in case of biological method. Because of that, I'm just coming to underline once again, biological method is not so simple compared with the chemical one. So, at this moment, we have information about the characterization of metallic core, proving of such point, screening that we have or not organic deposit. But what is next? The next, it is interesting which kind of organic deposit we have. So in this context, we are using laser distortion ionization time-of-flight mass spectrometry. Firstly, we are performing MS spectra. Then we select the characteristic signal, fragmentate this, and then identify it according to the database. And for example, here I have presented the results regarding the silver nanoparticles. But if we will compare the zinc oxide nanoparticles, we also will be able to identify firstly the amino acid sequences, and we can compare the organic surface from the silver synthesized by biological method, and we can see the differences compared with the chemically synthesized one. These organic plots will differ from one to another. What is interesting, if we will use different concentration, these sequences, this organic part, will differ from one concentration to another. If we will compare the zinc oxide nanocomposite synthesized by two different bacterial strains, we also will recognize and we will identify that this amino acid sequence differs from one bacteria to another. This aspect is connected with the secretion of each bacteria of the metabolites that then are involved in the synthesis of zinc oxide nanocomposite, but also in the silver nanocomposite using biological methods. We can see that organic quads play a crucial role and can vary from one concentration to another, from one bacteria to another. Let's move to the next step and let's discuss a little bit the impact of the concentration. It is well known that when we will have low concentration, we will have high dispersion of the system. With increasing of the concentration in case of biological method, our nanocomposite can agglomerate, and of course this is because of the increasing of the interparticle affinity. This aspect is correlated also with the stability property, means zeta potential value. Moreover, taking in consideration that our nanocomposites are as a biocolloids, in different pH, we will have different stability. This is one parameter that pH will influence the stability of the system because we will have different affinity between organic deposits. But we have also to keep in our mind that not only pH will influence the stability, but also the concentration. When we will have to compare, for example, several concentration and to biological synthesized nanocomposite and chemically synthesized nanocomposite, of course, we will see the differences. First, between chemically synthesized and biologically synthesized, but also we will see differences and strong differences between the used concentrations. Also, we have to keep in our mind that the concentration also is correlated with the size of bio-nanocomposite. When we discuss in context of zeta potential value, we discuss about the stability and size that was measured in hydrodynamic model of our nanocomposite. It's not silver nanoparticles. It is metal quatted together with organic quads. So, at this moment, we see that the size are correlated with pH, concentration, and this aspect, concentration, can influence also the stability. The concentration can also influence the fluorescence property. How we can remark here, at lower concentration, higher concentration, we did not receive high fluorescence property. The optimal concentration in our case, it was 250 and 500 microgram per milliliter in case of the zinc. In case of silver is different one. What I would like to underline here, that concentration is one aspect. Second one, it is differences between the nanocomposite synthesized by biological method and the nanocomposite synthesized by chemical one. What I would like also to specify that we remark also differences between the nanocomposites synthesized by biological method. This is proving once again that these organic quads influence just the fluorescence and proving that we have present in such concentration such organic surface, means individual one. So, at this moment, we saw concentration is directly correlated with the organics that is identified. Also with the fluorescence, concentration will influence the dispersion. As a consequence, the dispersion will influence the stability. All of these parameters put together will influence, of course, the bioavailability. It's interesting what is next. So, generally, I'm using two methods for the investigation of effectiveness of such nanocomposite. First one is the minimum inhibitory concentration method, that is standard method recommended by the clinical and the laboratory standard institution guidelines. Second one, it is flow cytometry. And I'm using as an alternative method in order to investigate the effectiveness. I said something about the flow cytometry before, and let's now to pass further and say something, some few words about this technique. This technique consists in passing of the samples through the capillary. When the sample is passing through the capillary, the cells are interrogated by the laser beam, also by the fluorescence detector. The data are received by the computer, and computer just converts the data in a dot plot or in form of the histogram. Also, when the sample is passing through the capillary, the data are interpreted during the time and are converted to the voltage. Also, when we have force scatter detection, our cells will be represented in the plot independent of the size. When we have side scatter detection, our samples will be represented in our plot independence of the granularity and complexity of our cells. More complex structure, it will be placed in this area of the plot. Lower complexity will be placed in such way. So, using two lasers, forward scatter and side scatter, we can represent our sample in the plot based on the size and the complexity. Also, we can use one fluorophore to identify it. Simultaneously, we can use two fluorophores to investigate our samples, and we can use three fluorophores and more, but we have to keep in our mind about the emission and excitation factor, of course. One aspect that today only I will specify, it is acquisition of threshold. The question it is, what is threshold in flow cytometry? The answer is simple. We just eliminate from our plot the cells that are not of our interest, and then we are focused only on the cells that we want to investigate. This acquisition we can do before analysis or after analysis. It depends on us. So let's back to the concentration impact. Here I would like to discuss the concentration impact versus antimicrobial effect. I have represented here pure concentration of silver nanocomposite. We can remark one aspect. With decreasing of the concentration, the light cells just increase. When we choose, for us, in this case, it's lower concentration, higher concentration. When we are choosing the optimal concentration, we can combine such nanocomposite with antibiotics and investigate the synergy. But in case of another bacterial strain, we can obtain different trend. For example, when we will use the same nanocomposite, the same concentration in case of another microorganism, we observe different trend with increasing of the concentration, increase the number of dead cells. Then combining this nanocomposite with antibiotics, we are able just to recognize which combination can be used for the inhibition. So using such technique, I investigated separate bio-nanocomposite with different concentration, also different antibiotics with different concentration. Generally, we are focused more on the mean non-density concentration of antibiotics, but in case of nanocomposite, when we want to combine both of them and obtain such formulation, always, firstly, we have to perform investigation of various concentration to find the optimal concentration that will be used for the combination with various antibiotics. What I would like to specify here also, how this concentration and not only influence the antimicrobial effect. Please put your attention here. We have the same nanocomposite combined with two different antibiotics. Of course, we will have different concentration of antibiotic. We will have the same concentration of nanocomposite, but we receive different antimicrobial effects. Here we play the crucial role, not only concentration, but also how this functional group that are coming from the antibiotics are binded to the group, to the functional group that are coming from the nanocomposite. And this binding also will decide the antimicrobial effect. So then combining such approach, flow cytometry with laser distortion ionization time of flight mass spectrometry, we will be able to design the mechanism generated by such complex that is more effective, and the mechanism generated by the such complex that is less effective. So at this moment, we have such information that the concentration is connected with stability and also with the effectiveness. What I would like to specify here in this slide, I would like to discuss also impact of the concentration versus antimicrobial effects and versus nanocomposite type. I have investigated the same bacterial strain, different nanocomposites, silver nanocomposites, six samples, and I have also investigated 3 types of nanocomposites, zinc oxide nanocomposite, but at a different concentration. From the minimum inhibitory concentration, it was found that zinc oxide nanocomposite present higher minimal inhibitory concentration compared with the silver nanocomposite. But what is more interesting here, it is that, firstly, we can observe the differences between the concentration, between effects and or antimicrobial effect of the concentration generated by different compounds, silver nanocomposites. Different effects against the same bacterial strain will give by the zinc oxide nanocomposite. And inhibitory effects, number of live cells, number of dead cells, also differ from one type to another, from one concentration to another. So, additionally to the concentration, stability, and effectiveness, crucial impact also will have property of the nanocomposites that we are using. Let's move now and let's convert our data from vitro to vivo. How we prepare such experiment? Firstly, we took all of the minimal inhibitory concentration from all of our samples that we received from the flow cytometry and standard method. Then I was responsible for the preparation of such ointment. Such ointment contain all of our nanocomposites that I was discussed today with the optimal concentration, means minimal inhibitory concentration that we found that it is enough to inhibit the bacterial growth. What is next? From the mice who were collectively liver and skin, this part of the experiment was performed by my colleague using ICP-MS technique. I collected just blood and used flow cytometry for the identification of the cytotoxicity effect of our ointments that contain such nanocomposites. In this case, we use several nanocomposites. As a control was one antibiotic as well. In this case, we investigate meta-proteins as well, zinc and silver nanocomposites. So let's discuss the results. Observation was very interesting and very surprised for us, of course, because in all cases of our nanocomposites was found the alive cells more than 97%. It means that this nanocomposite with such concentration, minimal inhibitory concentration, is not toxic for our mice, but it's toxic and it is enough to inhibit the bacterial growth. This aspect, it was a very interesting conclusion for us and hopeful for the future for applying for other projects. So summarizing all of my talk today, I would like just to underline a few aspects shortly. Firstly, that this talk is planning to promote the recycling approach using lactic acid bacterial strain, for example, for the synthesis of silver or zinc oxide nanocomposites. Secondly, this work pursues to design new complementary interpretation and to employ such complementary interpretation. In my opinion, it is a crucial point in characterization step, especially when we have discussed about the silver nanocomposite, pure silver nanocomposite, and silver chloride nanocomposite. Also, the present study is coming just to supplement the present knowledge regarding the bio-nanocomposite concerning structure, organics, stability of the biocolloids in the time, similarity, dissimilarity from 2 points of view. The bacteria that are used for the synthesis, and also the type, between the type of the nanocomposite synthesized by the same bacterial strain. What I would like also to underline here is the fact that during my experience with such nanocomposite, of course, the ecological problem just encouraged me and also my group to design such complex interdisciplinary analysis that will give a new scientific approach concerning the nanocomposites that are synthesized by biological method, and especially nanoparticles synthesized by biological method that are naturally coated by the organic core. Thank you for your attention.
Thank you for a very interesting lecture. Do any of you have any question? Question can be asked in both language, English and Polish. So
Tak, oczywiście.
we wait for any question.
Ja chętnie oczywiście odpowiadam.
Okay, so when we have a question, I want to thank you once again for an interesting lecture, and I invite our speakers to individual panel. Thank you so much.
Thank you very much for the invitation.